Hello there, how are you? It's been a long time since I've posted a video, and I thought we'll start back up with one of my lectures that I recently did for a group called Sisters in Crime. They're mystery writers, so it's super cool how they actually come up with their concepts and how they start uh, writing their stories. They actually, this was the first time, I knew they did a lot of research. I did not know to what extent they learn about the material that they write about. So they had invited me to do a little talk on how familial genealogy and genetics are used to bring justice. So that's what my presentation was about. So I thought I would share some of that with you because it might be of interest to you as well. So what this is going to be about is how genealogy and genetics are used to bring justice. So what exactly is genetic genealogy? Well, it is when we use DNA sequence information along with, traditionally just when we use sequence information to find relationships between individuals and to see how they are related. It could be individuals, it could be populations, right? It could be across different organisms. It could be a lot of different things. But it is how when DNA sequence information is used to grow your family tree or your relationships with one another. In genetic genealogy that is used most commonly by DNA detectives, it's a little bit more than that. It's using traditional genealogical information from your DNA and combining it with historical records or other information about that person in order to infer relationships between different individuals and document evidence. So it can include taking the evidence they have outside of just the DNA sequence to make this relationship. Sir Alex Jeffreys is a British geneticist. He developed the original techniques for genetic fingerprinting and DNA profiling that are used by forensic scientists across the world to assist any kind of detective work from police to resolving paternity or immigration disputes, any such things that require DNA information. So before we can go into DNA genetic profiling, we first have to kind of go back and review what exactly is DNA. I know some of you will be quite familiar with it. Others may not be so familiar if you're not in science. So we are not into biology too much. Everyone hears about DNA, and they know kind of that it is kind of like your code of life or genetic information, but that is inherited, but you don't exactly know the ins and outs of it. So we'll start a little bit from there. So DNA is deoxyribonucleic acid. It is an information storage molecule. It's an organic chemical that stores information. In our case, information about who we are and what we are, right? So it's instructions on anything and everything our body needs to make, how to make it, what to do with it. And a lot of times these instructions involve building proteins of one sort or another that can do the work in our bodies. It is at the end of the day our code of life or a recipe book to make us who we are. When we think about what DNA is good for, other than us as individuals, it is actually the instructions within it is what you inherit and pass down from one parent to the offspring, from the parents to the offspring. So each individual will pass out half of its genome to its offspring, and the other half was going to come from the other parent. Now, this doesn't mean that, chromosome 1 to 10 is going to be from mom and 11 through 20 is going to be from dad. Nothing like this. It actually means that for each individual chromosome, so the entire recipe book has two versions in your body. One of them came from your mom and one version came from your dad. So it has every single recipe in two flavors. So if you think about a recipe like apple pie, right? It has the mom side version of it and it has the dad side version of it. Lasagna, it has the mom version of it and a dad version of it. Obviously, it's not recipes like that inside our body, but rather it is, eye color or ability to a better ability to do something, or height, or skin color, any of those things, hair texture, they are coming from both parents, not one specifically. How does DNA do this? Well, DNA itself is a molecule that consists of two complementary strands of information. Um, and that is done because there's a specific base pairing rule that applies in DNA. There are, just like us, when we write in the English language, there are letters that make up words. In DNA, there are letters that make up words. There are 4 letters only, however, in DNA. These 4 letters are paired a very particular way. G can only pair with C, A can only pair with T. So using that restriction, the sequences can be made to make different proteins and different things. The shape of the DNA is like a twisted ladder. So it's two complementary strands interacting with each other and then twisted up. And this particular shape is called the double helix. Watson and Crick were 2 scientists that were doing their PhD in England when they first came up with the model, the correct 3D model for DNA structure, using information from a lot of other more senior scientists, including Rosalind Franklin's famous Photo 51, which was done with X-ray diffraction, that showed that this had to be a helical structure. So we, as humans, are considered eukaryotic organisms. That means our bodies are made up of cells in a very particular way, where the cell has every single cell has a central command center that houses the nucleus. That's the nucleus, and the DNA is housed within that nucleus of the cell. The DNA is protected in that environment, right? Because it's all the way inside the cell in the central area. And it's all packed in tightly within it so that it can fit. Only portions of it at any given time are opened up so they can be copied and made into proteins or made to use as needed. Human genomes contain 3.2 billion nucleotides, and they are packaged into 23 pairs of chromosomes. There is a typo here. I apologize. It's 23 pairs of chromosomes. These chromosomes are not in their linear separated form, but rather they are packed inside the cell as what we call chromatin, which is a complex of DNA and proteins. The proteins are helping to create a compact structure. They're like yo-yos, and the DNA is wrapped around them and then stuck together inside, packed together, tightly wound in the nucleus. This chromatin condenses into chromosomes, which are then inherited from your parents. When you have cells ready to divide, they get into this particular state where the chromosomes are condensed and separated out. And if at that time you were to take those cells, fix them, and stain them with special dyes, you can create what we call a karyogram. Karyogram is an image where all the chromosomes are organized in a way that you can clearly see what is there, what is not, and make sure that everything is there and everything is in the correct, its normal state, that there is nothing broken, there is nothing lost or added, there's no swapping happening between different chromosomes. This technique is called is called chromosome painting, And this is called a karyotype. A karyotype is basically all your chromosomes together make up your karyotype. And a karyogram is the image that we get when we use this technique and organize them in this manner. This will also let you know if somebody that you are looking at is a boy or a girl. The first 22 chromosomes that we have are just our chromosomes that all of us have, whether you're a boy or a girl. The last pair is what determines our sex. If you have two X chromosomes, then genetically you're a girl. And if you have an X and a Y chromosome, then genetically you are a boy. Now, these genes are one thing that the genes in the nucleus are one thing that we inherit from both parents. In addition to that, there are other genes inside each one of our cells that are only carried through the mother or only passed on through the mother. And these other genes that are present in another organelle, another space within the cell, not the nucleus, which is called the mitochondria. Mitochondria is the powerhouse of cells and it helps to produce energy for our work. So it takes the food that we eat and it breaks it down to a form where it can be converted into active energy that can then be utilized by our cells for their work. Now, this mitochondria has DNA of its own. It's in a different shape. This DNA is circular, and you can have many copies of it inside each one of your mitochondria, unlike the DNA that's in the nucleus, which is only going to have two copies, one from mom and one from dad. Mitochondria mitochondrial DNA can be in multiple copies and only comes from the mom, nobody else. So it is a way to go through maternal linkage and look at the maternal lineage of the individual that you're examining. So here is an example. If you have, for example, this mom and this dad, all the kids will contain DNA from copies from mom and dad, but the DNA inside the mitochondria is only going to be from the mom. None of it is going to come from the dad. So the mitochondrial DNA is very small, very short, very small little thing. It's only about 17,000 base pairs and it contains 37 genes. But most of these genes are extremely essential. And a mutation in there or a problem in there can lead to actually a lethal response. It has a high mutation rate because it's not as protected as your typical nucleus, and it is in a place which contains a lot of reactive oxygen species that can help to cause more mutations as well. However, this is a way to follow maternal linkage or maternal lineage. So again, reminding that the nuclear DNA is inherited from all ancestors. Mitochondrial DNA is inherited only from the mom, single lineage, which comes from the mother. The mitochondrial DNA is another way that can be of a very important use in looking at DNA at crime scenes. Sometimes the DNA, the nuclear DNA, is too damaged or too degraded, or it's been so long in such harsh conditions that it is no longer detectable at a level where it can be useful. Even at those time, in those compromised samples, the mitochondrial DNA is a little bit more present. It's so small that it has a harder time getting taken away or getting contaminated, especially because it's inside the mitochondria. So the mitochondrial DNA typing can be useful in identifying people when you have bodies or samples that are not identifiable with typical techniques. It can allow you to identify remains that are upwards of 2,500 years old. So that's a very long time. case where we used mitochondrial DNA for finding the suspect, finding the criminal who did these rapes, was a case for about Gary Ridgway. Gary Ridgway was an American serial killer, and he was initially convicted of 48 separate murders between 1982 and 1998. However, he claims that he has killed as many as 80 women. He was arrested on a DNA match on a degraded body from one of these women that was able to catch them. And it was done using mitochondrial DNA typing, not the traditional DNA typing like we've seen before. I have a new mic and I'm trying it out today. So let's see. Our new webcam and mic. So not all DNA is packed the same inside the chromosomes. So the mitochondrial DNA is circular. It hangs out in the mitochondria. It does its thing. But the nuclear DNA is linear, and it is packed in differentially in the nucleus depending on whether it is actively used all the time or whether it's used only some of the time or whether it's never used. So there are two different types of DNA that you will find typically inside the nucleus. One is called euchromatin, and this is an extended, more loosely packed, accessible for gene expression form because it's going to be more open. The other type is heterochromatin, that's more tightly packed, so it is harder to get to it and it is not generally going to be expressed. These heterochromatin areas are not active all the time, but that doesn't mean that it does not contain important information. Sometimes they still have very important genes that are only transiently needed. So in addition to that, well, how do we control this, right? We control this by creating specific modifications on the DNA so that it is marked as either open or closed. There are several different types of modifications that can happen. You can have methylations, acetylations, right? Phosphorylations, or a combination at a given site. And typically, when it is just methylated, it leads to heterochromatin formation and gene silencing. So those areas are locked up. And if it is acetylated or phosphorylated, it will typically lead to a more open conformation where things are expressed. That doesn't mean that's always the rule. Sometimes it's a mixture. It's methylated and acetylated, and it's still getting expressed. This type of DNA methylation patterns are actually very faithfully inherited between parents and their offsprings, and it leads to imprinting certain properties in the next generation. Here is an example where we're looking at 3 alleles. In one parent, the mom, she has the allele. Allele is basically the flavor of the recipe that you have, right? The type recipes subtype. So the first allele, allele A, mom has the one that dislikes spicy food versus dad who likes to eat spicy food. The second allele they have is a normal risk for cancer, and dad instead has high risk of cancer. And then finally, the third allele they have, the mom has proficiency in crossword puzzles, while dad is not especially good at it. Having said that, the male imprinted gene, the not being able to play crossword puzzle is imprinted, which essentially overrides it, so it doesn't really work. And in mom case, normal risk of cancer is imprinted, so it doesn't really, it's not really effective, and it still gives her a higher case, level of risk of cancer if that's what she has on the other side. So these imprints are going to faithfully pass on to the kid when they get that copy. So this is the copy that the kid is getting from the mom, from the egg, and this is the one that's coming in from the dad, from the sperm. When they combine together, in the first instance, the mom had disliked spicy food, dad likes spicy food. Neither one of them are imprinted, but dislike of spicy food is the more dominant one. So that's the one that's going to be expressed. That's the kid is not going to like spicy food. In the second case, the mom had normal risk of cancer, but the dad had high risk of cancer. Unfortunately, mom's gene for low risk of cancer or normal risk of cancer is imprinted. So it is, even though it was the dominant type, because it's imprinted, it's not going to work. And the kid is going to be born with high risk of cancer. And lastly, mom had proficiency at crossword puzzles. Dad did not. Um, dad Dad's again allele was dad's flavor of the recipe was more prominent or more dominant, but it's not going to work because it was imprinted, which means the kid is going to be proficient at crossword. So looking at these imprinting patterns can help us figure out what the next generation may look like based on that. Another way that this kind of gene expression modeling happens is in X inactivation. So in females, you have two X chromosomes, while in males, you have one copy of X and then a Y chromosome that makes them the male, hormones and male genes. Now, the X chromosome, if you're a female, you don't need, it has a lot more genes, right? It has 1,000 genes on it. And so you don't need all of those genes in double dose. You want to still balance out the amount of that gene expression that's happening. So in every single cell of your body, one of the gene is going to be one of the X chromosome. The entire X chromosome is going to be randomly methylated and shut down. Because of this inactivity, inactive X chromosome, your cells, all of your cells will only express the right amount of the gene, will make the right amount of the recipe for each of the X genes. However, because it was randomly inactivating one or the other, about half of your cells are going to be making one type of recipe, one flavor, and the other one's going to be making the other flavor. That means that every single female, whether or not you can physically see that, is a mosaic, where half of your cells have some genes that are different, differentially expressed than the remaining because they happen to be on those X chromosomes and they were differentially switched off depending on which one was which. And so those are two good ways that you can follow lineage and follow which type of genes are expressed or not expressed. Now, finally, I've been talking about genes, but I never really explained what they were. Genes are your individual recipes, essentially. It's a segment of DNA that contains instructions for making a particular protein working molecule or an RNA molecule. And that's what we are finally going to be working with. On the other hand, genome is all your genes combined together, all the genetic information present in an organism in the form of its complete set of DNA, all two copies combined. Most of your genes are going to be coding for proteins. So from each of those recipes, each one of those genes, you're going to get one protein out of it. It's going to be for a specific function, for a specific job inside your body. Gene mapping is a way that we can examine the entire genome and map out which gene was gotten from which parent, where it exists on the genome, which chromosome next to what. And because by mapping them on these chromosomes, we can look at possible disease transmission from the parent to the child when it's linked by one or more genes. We can also similarly look at traits that are being passed from one parent to another based on the presence of particular genes on a particular space. So that is the basis for genetic genealogy, all this information that we just talked about. And it is us then using this information in the form of DNA profiles, DNA sequence information to infer biological relationships between individuals. Now, this is become very, very prevalent, very affordable, and that is why it has been, because it is affordable and very easy to do. Now we can actually get meaningful information from it and actually make it useful in forensics and in genetic analyses. A interesting tidbit is that the worldwide genealogical records are held by the Church of Latter-day Saints in Utah. I found that very interesting that every single gene sequence that has ever been made from, DNA profile that's ever been made from any of the humans, DNA analysis are all held by this one organization. Today, we have lots and lots of websites where you can privately get this information or where you can, by paying for them. There are other places where you can get them done as well. These websites include Ancestry.com, 23andMe, Family Tree DNA, and GEDmatch. GEDmatch actually doesn't perform the DNA analysis itself, but it is an open source. The other two are closed, right? Ancestry.com, 23andMe, and Family Tree DNA are all closed websites. You have to pay for them to do the service, and then they have your profile on file. Nobody can go get it without permission from you. But if you want yours to be part of the open space where anyone can get to it, anyone can see it, then you would want to download that copy from that website and upload it into GEDmatch. GEDmatch is what is used by genealogists and DNA forensic artists and everyone to find culprits, to find all kinds of information about people, to find ancestry roots. It is open source. It does not, it's basically you are giving them permission to share that information when you upload your sequence to there. And it has a vast array of tools to determine ancestry of people and look at anything like that. It is said that Confucius had a family tree that extends back 5,000 years. But now with DNA technology, not only can we find this information, but we can also look at how our ethnic percentage, heritage by percentage point, what percentage of our DNA came from, what lineage over time, over all these hundreds of years. So genealogy has become more than just the study of old bones and bloodlines. It actually gives you a lot more information and it has useful applications, including bringing long delayed justice for thousands of people. How exactly does it do that? Is going to be in the next part of my lecture that I will post later. So stay tuned for that and I will talk to you later. Take care. Bye bye.